Everyone. So in terms of timeline, I'm his recent postdoctoral fellow. And before starting, I would actually render my thanks to him because he has given me complete freedom to explore the topic that I'm interested in. And I'm somebody who takes up a lot of pressure, and most of it is self-induced. But his ever welcoming and infectious smile that we have all been talking about, puts me on ease. Yeah. So today I'll be talking about cellular plasticity, mostly in the context of melanoma, and a recent, which was one of the topics that I worked in my PhD, and more insights into hepatocellular carcinoma, which I'm currently working with. So cutaneous malignant melanoma that arises from this fully differentiated pigment-producing melanocytes is highly aggressive and heterogeneous cancer, quite infamous for its invasive potential. And heterogeneity can be intratumoral or intertumoral, having distinct subpopulations, each with their characteristic molecular profiles and functional attributes. They might have differing proliferation rate, differing invasive potential, and all of these can have their underlying genomic and non-genomic basis. But for the purpose of this study, we specifically focused on the non-genetic phenotypic aspect of melanoma heterogeneity, and also in case of liver heterogeneity that arises as a result of proliferative to invasive transition in melanoma and epithelial to mesenchymal transition in the context of liver cancer. Previously, it was seen that the melanoma cells, they can switch from a proliferative phenotype to an invasive phenotype, aiding in drug tolerance, immune evasion. And certain people have categorized these cell phenotypes into 4 types. So broadly, 2 bins, proliferative and invasive. And in proliferative, you have your melanocytic or proliferative, intermediate, and in invasive, you have your neural crest and invasive population, of which is the most undifferentiated subtype. So this happens by the induction of your dedifferentiation program. Now, what we wanted to look at is how do these phenotypically plastic states in melanoma arise? For that purpose, we looked at gene regulatory networks. So we looked into literature and basically came up with a very small minimalistic gene regulatory network consisting of transcription factors, which are commonly implicated in this process of proliferative to invasive transition. We incorporated the literature-derived regulatory links among them, and then simulated the entire gene regulatory network as a couple of, as a bunch of ODEs. So the way we write is, for example, if you have like a toggle switch, so A inhibits B, B inhibits A, then you can have a production term, you can have a regulation term, and then you have degradation terms. We use a framework for this. I'm going a little fast because I have to cover, but you can always ask me, stop me, and or we can catch up during lunchtime. So the framework we used is RACIP, which is random circuit perturbation. So we can vary the initial state of each of these transcription factors and also vary the parameter regimes. For example, we can vary the Hill coefficient, the degradation rates, the production rate, and like that. If you look at the steady state solutions varying all these parameters, you look at and then just arrange it as a heat map and do a hierarchical clustering, you can see that some of the transcription factors, they co-express together, for example, MITF and SOX10, and some of the other transcription factors, they are co-expressed together. So what you can see is typically this gives you 4 clusters. So the green cluster where you have your MITF as high, your SOX, so now you can just binarize their expression based on highs and lows, ones and zeros. So if you look at the then you project all of these solutions in a proliferative invasive space. So if you look at the green cluster, which is high in MITF, which is high in SOX10, that's the most differentiated phenotype, or that's more proliferative. But if you look at the orange cluster where you have the MITF low, SOX10 low, but the other 3 invasive master regulators on, which is SOX9, June, and ZEB1, you get something as an undifferentiated or most dedifferentiated cluster. So this is basically from the simulation, but it makes sense if you also see that in data. For that, we do single cell transcriptomics, where if you look at these patient-derived melanoma cell cultures, look at the expression of these transcription factors, and you see bimodality. At the minima, you can split them as ones and zeros, and you can see that all majority of the cell population in each of these cell lines actually have a phenotype that is described by the network and also previously identified in the literature. What is interesting is you come across these phenotypes, which is sort of an intermediate or neural crest phenotypes, which does not become intuitive from the network, where you can see that some of the programs, for example, SOX9 is sort of, this cluster basically. You look at SOX10 is high, SOX9 and MITF are low, and the other ones could be high or low. So this is some sort of intermediate phenotype. Then what we can do is, so often in experimental labs, to test what happens or what can induce this proliferative to invasive transition, you can basically down-regulate or up-regulate a gene. But before testing it out experimentally, we can actually we can perform in silico simulations. For example, if I down-regulate or down-express one of my transcription factors, what happens? So you see that the proliferative population decreases, the invasive population increases, and the neural crest and intermediate show these type of trends. And then we can also see whether our simulation results are seen in experimental data. So for this, we analyze, so I think this was like bulk transcriptomics data, which had SOX10 down expressed, or there was basically an shRNA which stops the SOX10. Similarly, we can upregulate or overexpress any of the transcription factors and see the trends. To look at the single cell transcriptomics data, you see that the gray cluster, it's basically the wild type cluster. But as you perform a knockdown or a down expression, you can see that the basically phenotypic switching happening, where after 72 hours of SOX10 knockdown, you see a cell population which acquire a transcriptional state which is more invasive than previously to begin with. So basically, we can map out how this transition from a more proliferative to a more invasive population going on. Next, something that we were more interested in is looking at combinatorial therapies, looking at immune checkpoint inhibitors. So for this, we saw that these phenotypic subtypes, they display varying immune suppressive or immune evasive capabilities. So we link PDL1, which is basically present in your cancer cells, which on binding to PD1, which are present in your T cells, lead to T cell exhaustion. So this is one of the markers to study, and then we connected it with interferon gamma, and then we performed the simulations that we were performing previously. What we saw is that the invasive and the neural crest, which is more dedifferentiated, they have like mid to higher level of PDL1, whereas the proliferative and intermediate have lower levels of PDL1. And this was also seen in data. So the more dedifferentiated a phenotype becomes, the higher is the PDL1 expression level, as well as the variation in PDL1. So there are like two components which we have added. One is PDL1 and interferon gamma. Now, interferon gamma also upregulates PDL1, and it is an independent parameter which also leads to differentiation. So I would like you to imagine two axes. One where you don't give anything, but the transition from a more differentiated to dedifferentiated phenotype happens via the proliferative to invasive transition. That's one of your axes, and the other axis is your interferon gamma. And both of these axes feed into the level of PDL1. And PDL1 is basically one of our markers to target, because you can give anti-PDL1 therapies for it. Now, how do this two axes crosstalk with PDL1? And which axis has a higher contribution is something we wanted to know. So we simulated two scenarios. We splitted the phenotypes into a lower interferon gamma regime and those in the higher interferon gamma regime, and we looked at the PDL1 levels. So if you focus on the low interferon gamma regime and look at just the PDL1 levels, you see that the more differentiated phenotypes, the proliferative and intermediate, they have lower levels of PDL1, whereas the more dedifferentiated phenotypes have mid to higher range of PD-L1. But the moment you shift to a higher interferon gamma regime, even the basal PD-L1 levels of all of these phenotypes sort of increases. And this is actually, and obviously the higher being in the most dedifferentiated phenotypes. And this can also be seen in data. So this is basically a single cell transcriptomics data of immunotherapy. And if you look at just the blue cluster, which is a high cluster, which is more invasive and has higher interferon gamma, that has the highest level of PD-L1. And if you look at the pink cluster, which is a low cluster, which is more proliferative and has lesser interferon gamma, that has the least PD-L1. So right now we know that, okay, interferon gamma has a higher contribution to the effect of PD-L1, but on combination, you see that there's just the highest level of upregulation of PD-L1. And we went ahead, and with the help of our experimental collaborators at Duke University, we sort of tested this hypothesis. Now, we treated the cells with TNF alpha and TGF beta, which will induce the cells to become more dedifferentiated, and you treat separately with interferon gamma because it also helps in dedifferentiation, and then you just have a combinatorial treatment. And you see that in the combinatorial treatment, there is like a very high upshooting of the PD-L1 levels. For the interest of time, I'll not go into the drug resistance, but happy to chat about it in terms of targeted therapy and immunotherapy, but I'll just switch on to a liver. In the context of hepatocellular carcinoma, so the way melanoma and hepatocellular carcinoma differs is because liver cancer is a solid tumor. So you see the typical effects of epithelial to mesenchymal going on. Whereas melanoma is not a solid tumor. So the phenomena is slightly different, but almost synonymous, where there is this proliferative to invasive transition. And HCC is one of the most common type of liver cancers which happens in adults. And the problem with HCC is it has very nil to minimal symptoms. So it's often quite late when the cancer is sort of detected. It's usually at an advanced stage. And because heterogeneity possesses a major challenge, and in terms of liver cancer, not only heterogeneity in terms of molecular signatures, but also etiology, because people who have long-standing infections of hepatitis B and hepatitis C can develop liver cancer with cirrhosis, with alcohol consumptions. All of these can lead to liver cancer. And there are many groups which have come up to basically understand how you can stratify the HCC into different subtypes with the help of unique biological characteristic, clinical outcomes, and therapeutic outcomes. One of the groups is HOSIDA, which have 3 subtypes of HCC, and then they look at the EMT characteristics, the immune subclasses, and similarly the one et al, which looks into 5 subtypes. So I'll not go into much of the details in this presentation, but what we have seen is we see a very antagonistic relation between the liver phenotype as well as the mesenchymal characteristics. So when I say liver phenotype, it's a more differentiated phenotype. So you see that there is an antagonism between hepatocyte score and EMT, which gives you a status of more mesenchymal subtype. And if you just club all the transcription factors, the top transcription factors in each of these programs, you can see that there are 2 teams of players and there is clear antagonism between them. If you look at the survival data, the adult liver signatures is predictive of better survival in both the overall survival as well as the disease-free survival. So the red curve is like higher, and higher is your adult liver signature, or more differentiated is the phenotype. It has a better survival prognosis. Similarly, the higher EMT has or more dedifferentiated a phenotype is it has lower survival. And if you look at just the transcription factors and the role of both these signatures, you see a clear antagonism. This is not the final gene regulatory network, but the purpose of this talk, I just simulated something based on the few markers that I was looking at. And even if you just take these few markers, you can see that there are clear populations that are emerging, one which is more mesenchymal and one which is more differentiated, and you have those hybrid clusters. Still, the work is in progress. And I would like to thank my collaborators, Professor Sivalokanatham, Professor Mohit Kumar Jolly, and Sarthak for this.
Thank you very much, Seemadri.
I think